Hi, everyone. Thanks for being here. Once again, my name is Leah, and I'm a fourth-year graduate student in the Dunham Lab, where we study Saccharomyces cerevisiae, which is one of the model organisms that Riku introduced. I like to illustrate them as this little thing here, so you'll see this throughout my talk to represent the yeast that I'm working with. And basically, my project is generating freeze-thaw tolerant yeast and using genomics tools to figure out what caused them to be freeze-thaw tolerant. So this organism, Saccharomyces cerevisiae, is a great one to know. If you're interested in genomics or microbiology or most life science fields, it's going to come up in the research because it's just so widely used for a lot of great reasons, mainly the ones that Riku introduced. But it's also really cool because it's used for a lot of other applications outside of the lab. So a lot of you might have used this species to bake bread at home or other baked goods. We also use this same exact species to ferment beer, wine, and coffee. A lot of people don't know coffee beans are fermented by yeast too. And also to produce biofuels too, so that it has a place in kind of the sustainability realm as well. And this is just the tip of the iceberg. Saccharomyces cerevisiae is used for so many different things. And across all of these diverse applications, the most common way for people to store their specific yeast strains that they're using is by freezing. So people will take a liquid culture of the yeast, combine it with a cryoprotective chemical, and store it in anywhere from -20 to -80 degrees Celsius. But even though this is a very common practice, freeze-thaw survival, depending on which strain you're using, it's never 100% and usually not even close to 100% survival. Because freezing is hard on an organism, as we can imagine. I know none of us would want to be stepping into a freezer for too long. And even though we do it all the time, we still don't know a lot about the genetics of freeze-thaw tolerance. So this is kind of where my motivations for my project come in. And I also want to share kind of a more personal anecdote, I guess. So in the Dunham Lab, we actually collaborate with a lot of small craft breweries. And during this heat dome event that some of you might remember in Seattle from, I believe that was summer of 2021, really intense heat wave, a lot of our collaborators at these small breweries, a lot of their yeast that they used to ferment their beers died. And so with the remaining yeast that they had that were somehow able to survive that heat wave, those couldn't even survive being stored in the freezer. So kind of, because it was kind of like a total loss of all brewing yeast during that really, really hot week. And so as we're dealing with more effects of climate change, it's becoming increasingly important for us to understand how we can manipulate this very important yeast to kind of be resilient to these, drastically changing temperatures. So yes, that it was definitely very motivating for me to see this firsthand. So the approach I've decided to use for studying freeze-thaw tolerance in yeast is experimental evolution. And experimental evolution is basically what it sounds like. You basically are using the power of adaptation and natural selection, but in a controlled laboratory setting. So basically you're exposing your organism to an extreme condition and basically allowing the survivors to grow. So in this case, it'd be these cycles of freezing and thawing, which is the extreme condition. And then the ones that are able to survive that will then continue to propagate and bud and have more offspring. So over the course of the experiment, as random mutations pop up, and this is just, all organisms have random mutations. It's just a part of life. Any of these random mutations represented by stars here, if they confer a fitness benefit, would see them sweep the population or rise to high frequency. So by the end of the experiment, we'd have these mutated yeasts that are ideally more freeze-thaw tolerant than the starting population and have some genetic changes that we can then measure. So that was the idea with this. And so I went ahead and designed this freeze-thaw evolution protocol, and I conducted these experiments with the help of a talented undergraduate, Leah. And basically what we did was we started 32 independent freeze-thaw experimental evolution experiments. And each of these were started from a single yeast cell. So the starting population was all clones. So every cell in the culture was genetically identical. And then we put them through these freeze-thaw events. So this is being frozen at -80 degrees Celsius with no cryoprotecting chemicals. So it's very extreme. Most cells die in the early stages. And then we we do take the surviving ones and grow them back out. And over multiple cycles, we started to see that more and more cells were surviving. And we carried these out for 20 total experimental evolution cycles. So one full cycle being from here all the way back to this stage. And then we also designed an experiment to test how much more tolerant our strains were becoming. So basically what we do here is use this 96 well plate, and we can put 96 different yeast cultures into each of these little wells. And we can take that whole plate and freeze and thaw it multiple times. And then over the course of those multiple freeze thaw rounds, we can see which strains are able to survive more and more of those events. And we do this by using this device called a pinner. So basically it just has 96 little metal pins. You can stick it straight into the culture and touch it to a petri dish. And any well that had surviving cells in it will have a little colony that forms on the petri dish. So that's how we're able to tell which ones were surviving. So kind of what this looks like. So here's a control experiment, for example. So this was a 96 well plate that had the same strain all across the plate. So all of these wells just have our basic lab strain that was the same as the ancestor we used in these freeze thaw experimental evolution. And over the course of 2 to 6 freezes, we saw total loss of viability. You can see by freeze 5 here, there's only a few cells that were surviving. And so then what we can do is put both the evolved populations and the ancestor on the same plate. And we can see from freeze number 2, where everything is still surviving, to freeze number 5, those rows that had the ancestor in it are totally dead, whereas the evolved populations are now able to survive more and more freezes. And you can plot it as a survival curve too. So here I'm showing like the different stages of the experimental evolution. So cycle 0 being the ancestor, and then 5 and 10 being after 5 and 10 evolution cycles. We can see that those populations are surviving more and more of those freeze thaw events, which is very promising. And then when we look at cycle 20, some of them were really extremely different from the ancestor, surviving as many as 14 freeze thaw events. So this was very exciting to see that our evolutions worked the way we expected. And so then the next step was, how do we detect the changes that occurred? And so this is where we decided to do next generation genome sequencing. And this is like very central to what we do in genome sciences. I would guess pretty much every lab here does this in some capacity. So I just wanted to outline how it works. So basically you extract whole genome DNA from your organism. So in my case, I have an ancestor involved yeast, and you shear it up into a bunch of small fragments, and then you can sequence those millions of fragments on an Illumina sequencer. So this is like a company, Illumina, that makes these devices. And then you end up with all these millions of sequence reads, and we can use computational pipelines to match up those sequence reads to the reference genome. So that's what I'm showing here, is those mapping there. And so then this is what my actual data looks like. So this is like a software that we can use to visualize those reads. So these gray bars are all reads that map to the genome. The genome is right down here, so you can see all those base pairs. And anytime the bar here is gray, that means it perfectly matches to the genome. But anytime there's a colorful nucleotide, that means it doesn't match. So here we're seeing an example of our evolved sample that's differing from the reference genome and the ancestor. So we're saying, okay, here we had like 90% of the reads are showing a G instead of a T, which might indicate that this is beneficial for surviving being frozen. So using this method, I was able to identify 23 different genes that were mutated in my many evolutions that I did. And what I'm going to show on the y-axis here is how many independent populations had mutations in these genes. So the more populations that had mutations in those genes, the more confident I feel that it could be causing freeze-thaw tolerance. And I was excited to see that some of the genes we hit were previously cited in the literature to be involved either with freeze-thaw tolerance or related phenotypes. But surprisingly, I actually saw a lot of genes that had never been connected to freeze-thaw tolerance in the past, including these 4 at the front here that had multiple populations that they were mutated in. So this is very exciting to see. And of course, I wish I had time to talk about all of them, but I don't. So I'm just going to focus on these 2 here, KEX1 and 2, because they're related to each other, like they're orthologs, and also they were both mutated a lot of times. So they're clearly important. So KEXins are these proteins called serine proteases, and they're basically involved with processing other proteins, and they're involved with all these signaling pathways in the cell. And I was also excited to see in the literature that there have been a couple studies that have connected these genes to heat or cold tolerance. So that was also promising. And then my next step here was to figure out what types of mutations occurred, because that can also tell us or give us hints as to what's happening mechanistically. And I noticed that most of the mutations we saw were SNPs. So this means a single nucleotide polymorphism, just one change, or an indel, so a small insertion or deletion. So this is what these would look like, just changing an A to a G or inserting an extra A. So that's like this picture that I showed you earlier. That's a SNP. But what was interesting about KEX1 is that, so again, here's the KEX1 locus. When I aligned this to 3 of my evolved populations, I saw a total absence of that gene locus. So this means that something happened in these evolutions 3 independent times where the whole gene was totally deleted. So the hypothesis here is that a total loss of function in this KEX1 gene causes free cell tolerance somehow. So this is like really exciting, and I'm going to be following up on this to figure out why. When we look at KEX2, it's a little less clear what the mechanism of mutation is, but we saw that most of them are missense SNPs, which means that just one amino acid was changed. So we are going to need further experiments to explore that and figure out what's going on. But something I did want to share, because I thought it was really interesting, is that KEX2 has a large interaction network, which is understandable as a serine protease. It processes other proteins. But what was interesting is that 2 of these genes in the interaction network were actually the other 2 that I saw recurrently mutated in my populations. So this means that there could be some sort of genetic network happening surrounding KEX2 that can be altered in some way that can confer freeze thaw tolerance. So again, we're going to need to follow up this work with other experiments, but it's very promising to see these connections already happening in our data. And I just want to illustrate again, most of these were missense mutations in my recurrently mutated genes. So again, we'll need to follow this up with extra experiments. So what I'm going to do is CRISPR. And I don't know if everyone's familiar with CRISPR here, but basically you can think of it as DNA scissors. So you can basically cut out the gene that's already in the yeast and then replace it with something else. So here my pink gene is the mutant that I want to study. So you can cut it in, or cut the original one out and paste this one in, and then I can test freeze thaw tolerance here. So these are the follow-up experiments I want to do, with the single mutations as well as like combinations. But that's going to take me a while. So I'm going to leave you with one preliminary experiment here. One great reason that we can study yeast is that we have all of these tools that the community has made. So the yeast knockout collection is one example where people have made every yeast strain with every gene knocked out. So I was able to just go into this collection that already exists and pull out all the deletions of my genes of interest and test their freeze thaw viability to just kind of get an idea of what a knockout would cause. And I did indeed see that the KEX1 and KEX2 deletions do cause this significant increase in freeze thaw viability, whereas all the other ones either had no change or had decreased freeze thaw survival, which means that the mutations I got in my evolutions and all these other genes were likely altered function in some way. So that's going to require more follow-up. But I'm excited to try to piece apart those differences in future work. So to summarize, freeze thaw experimental evolution is a way that we can make freeze thaw tolerant yeast and also learn about the genetics of this trait. And we also identified these KEX1 and KEX2 genes, which are novel factors in freeze thaw tolerance. And they seem to need to be a loss of function to cause that tolerance. And then in the future, we're hoping to validate the impacts of all these mutations, and then maybe even in the distant future, engineer strains like brewing strains that can be more resilient to being frozen and also tolerate the heat waves that we're dealing with, et cetera. So with that, I want to thank my lab and the organizers of this event, especially Stephen and Renee. And I'll take any questions, and I'll also be at the reception if people have questions there too.